Hey, what's going on, everybody? We are back for a third and final video on the brain. So we're going to talk about the cerebellum, then some other brain systems, and then we're going to talk about brain protection, and then we will be all done. So the cerebellum, which is in that black circle right there, it is inferior to the occipital lobe and it is superior to the, or sorry, posterior to the brain stem. So let's talk a bit about that cerebellum. It actually has two hemispheres. We see a hemisphere on one side, a hemisphere on the other side, as well as a vermis in between them. It also has three nodes. It has an it has sorry it has an anterior node, a posterior node, and this little guy called a flocculonodular node. So three basic nodes to it. Okay. Now, if we slice our cerebellum, we do see like a cortex of gray matter and then the internal white matter, which is the arbor vitae. The white matter, of course, is going to connect the cerebellum to the brainstem, to the diencephalon, and to the cerebrum. Now, the big functions of the cerebellum include motor processing, coordinating movement, balancing. It does play roles in languages, learning, emotions, and more. Check out how you have a little homunculus, a trio of homunculi in the cerebellum as well. Because just like we had maps of our body in our pre and post central gyri, we also need maps to the body in the cerebellum. Now, that's pretty much it for the cerebellum. Wow, that was really, really fast. Next up, we got something called the reticular formation, which is a group of interconnected nuclei in the brainstem. It's going to function in sleep-wake cycles, consciousness, attention, wakefulness, behavioral arousal, more like posture, emotions, all sorts of things. So it's basically a way for different brainstem structures to talk to one another and then to talk to the cerebrum, as well as get input from the body. All right, the final thing we're going to do before we get to protection of the brain is the limbic system. So the limbic system is this ring of structures involved in emotions, memories, behaviors, and kind of surprisingly, smelling or olfaction. There's some things we met already that are part of the limbic system, like the thalamus, the hypothalamus, and the fornix, but also some new things like the hippocampus, the cingulate gyrus, and the amygdala. So let's go through these new things. Now, the cingulate gyrus is a gyrus that is superior to the corpus callosum. So if you find that corpus callosum, you are going to find the cingulate gyrus around it. The word cingulate means like belt or girdle because of the way it kind of like surrounds the corpus callosum. It is very much involved in your emotional state. It is very much involved in behavior and memory as well. And these are all limbic system group functions. Now, the hippocampus is tucked in the medial temporal lobe, like kind of snaking. We see like the bottom of it right there, and it kind of like snakes up. The word hippocampus means seahorse because if you dissect out the hippocampus, someone thought it looked like a seahorse. I guess if you use your imagination. Hippocampus is very involved in long-term and spatial memory. Spatial memory. If you know about the mnemonic device, a memory palace. Classic way to remember things that involves your hippocampus. If you don't know about that, ask me. I will be glad to explain it to you in person. So that's my hippocampus. The amygdala is this little almond-shaped structure, also in the temporal lobe, near the hippocampus. Very much involved in emotions, decisions, memories, but also fear. Also fear. So that's my amygdala. I think the word amygdala means almond-like. I'm pretty sure it does. Yeah, it looks like an almond, so it's almond-like. Pretty small then for some important roles here. It's funny how like some of these brain structures are so tiny yet they have these just outsized roles. Anywho, the fornix, we met the fornix already. It's going to link your hippocampus to your thalamus and to your cerebrum. So this is a big job of the fornix, which we see right there, kind of below the corpus callosum, septum pellucidum. There's my thalamus right there. Okay, and with that, we're done with like actual brain structures, and we're moving on to brain protection. The brain is protected by bones, by meninges, and by cerebrospinal fluid. In this slice here, I do see bone. I see parietal bone right here. Then I see a bunch of meninges. I got 4 different meningeal membranes in this particular spot here. Then my cerebrospinal fluid would be in this space right here, marked by the blue arrow. Now, of course, the brain is in the cranial cavity. We met the bones of the cranial cavity previously. The frontal, the parietals, the temporals, the occipital, the sphenoid, and the ethmoid. So we see the temporal, the parietal, the occipital, the frontal, the sphenoid, the ethmoid. We can see them all here, all protecting the brain. If we slice that skull again, we see the frontal, we see the ethmoid, the sphenoid, the temporal, the occipital, and the parietals are kind of sliced off here. All right, that's the bones. We did those already. The meninges are a trio of membranes that anchor, protect, and cushion that brain. From outside to inside, there is the tough dura mater, the web-like arachnoid matter, and then the skinny, delicate pia mater. Mater means mother. I think it's a Persian saying where if 2 things are like next to one another, one is the mother of the other. I don't really know exactly. Forgive me for that. Dura means tough. Arachnoid means spider-like. And pia means delicate or gentle. So the dura mater is my outermost meninx. Notice the singular for meninges is meninx. So around the brain, the dura mater has 2 cranial sublayers. There's an outer layer called the periosteal layer and then a deep layer called the meningeal layer. Sorry, I paused it too because I was interrupted and I forgot where I was. Lost my train of thought. I think I basically just said there are 2 layers of the dura mater. The periosteal, which is next to the bone, and then the meningeal, which is the deeper one. Structurally, they're both made of dense irregular connective tissue, although I'll have some like osteoblasts and things in this one that will be absent in this guy. Now, usually the 2 layers are right up against one another. It's just like fused together here. But in certain spots, the 2 layers are separated. And where they're separated, there is usually a blood vessel between them. This blue guy right here. This blood vessel is called a dural venous sinus. A sinus is a big thin-walled vein. So we're using that word sinus with a brand new definition here. Now, what the dural venous sinuses do is they actually get CSF. So there would be CSF in this subarachnoid space here, and old CSF gets absorbed into this vein here. This is how we get rid of old CSF. We're constantly removing old CSF. It ends up in these dural venous sinuses. So it's how we get rid of old CSF. Okay, deep to my dura mater is my arachnoid, which is my middle meninx. Notice the arachnoid has all these fibers connecting it to the pia mater. They crisscross, they get web-like, so hence the name arachnoid, which means spider-y. Now, the space under my arachnoid is the subarachnoid space. This is where I will find the CSF that surrounds and protects my brain, giving it some shock absorption. Do notice how portions of the arachnoid matter kind of bulge into the dural venous sinus? This is called an arachnoid villus. I see two arachnoid villi here. And like we mentioned a moment ago, this is where CSF is draining into the bloodstream. Now, my deepest meninx is my pia mater. My pia mater is right on top of that cerebral cortex. My pia mater follows every gyrus and sulcus in and out, in and out. And it is just a very thin, delicate, loose connective tissue. Not nearly as dense as my arachnoid, or nowhere near as dense as my good old dura mater. Okay, so those are my meninges. We mentioned CSF. We mentioned it was in the subarachnoid space. So the light blue here represents CSF. CSF is also found in my ventricles. Here's my third ventricle. My fourth ventricle is shadowed here. My lateral ventricle is shadowed here. And my fourth ventricle is down here. The CSF primarily functions in shock absorption. We generally have about 125 mils of it in our body at any one time. Now, every choroid plexus is making CSF, and we have a ventricle in every choroid plexus. We have 4 choroid plexuses. I see one in my third ventricle. I see one in my fourth ventricle. My 2 lateral ventricles would each have a choroid plexus as well. And my choroid plexuses make about half a liter, about 500 mils of CSF in a day. And then that CSF is going to be in all 4 ventricles, flowing from one to the other to the other. So here I've got my lateral ventricles. Notice how big and curvy they are. So I see my 2 lateral ventricles, one in each cerebral hemisphere. My lateral ventricles are connected to my third ventricle. Here's my third ventricle. The connection is an interventricular foramen. Third ventricle, of course, is within the diencephalon. Then my third ventricle is connected down to my fourth. Here's my fourth ventricle between my brainstem and my cerebellum. And the connection between third and fourth is this cerebral aqueduct here. So CSF is made in the lateral ventricles, flows into the third through the interventricular foramina. Then the third adds more CSF of its own, flows down through the aqueduct into the fourth. The fourth ventricle adds more CSF of its own. Then the fourth ventricle has a opening in the back middle of it called the median aperture, and an opening in each side called the lateral aperture on each side. And those apertures allow the CSF to get into the subarachnoid space, which then surrounds the brain. Now, of course, some CSF just goes straight down from the fourth ventricle into the central canal of the spinal cord. So in just another view here, lateral ventricles in each hemisphere of the cerebrum, third ventricle in the diencephalon, interventricular foramina connecting those, cerebral aqueduct to the fourth ventricle. Fourth ventricle connects down to the central canal of the spinal cord. And then I also am going to see my lateral apertures here, which allows CSF to get into my subarachnoid space. So we just see the flow here, lateral ventricle to the third, third to the aqueduct to the fourth, down into the central canal, but also out the apertures into the subarachnoid space, all the way around the brain and the cord, eventually up into an arachnoid villus, where the CSF goes back into the blood of a dural venous sinus. Now, this movement of CSF is powered by the sweeping of the cilia, which are on the ependymal cells, which are lining all of these spaces, ventricles, aqueducts, central canal. So anywhere there's CSF inside the brain and the spinal cord, it is going to be lined by ependymal cells. And their cilia are sweeping, and that moves the CSF. All right, the final thing we got to talk about in terms of brain protection is this thing called the blood-brain barrier. Now, of course, your brain needs a lot of blood vessels to feed it. It has lots and lots of capillaries. Most capillaries in the brain are covered by this thing called the blood-brain barrier, which prevents toxins, pathogens, bad stuff from leaving the blood and getting into the brain's interstitial fluid. So this guy down here is an example of a capillary, a tiny blood vessel in the brain. Notice that it has simple squamous epithelial cells creating it, and they're actually connected by tight junctions. And we remember tight junctions are the impermeable connection, so it is very hard for things to get through the tight junctions linking these epithelial cells. Then plus, I see one of my glial cells here. This is an astrocyte. My astrocyte has these processes. Then it has these little guys called perivascular feet that are just grabbing onto the blood vessel and further covering it, providing a further impediment to anything that's trying to escape the lumen of the blood vessel and get into the interstitial space of the brain. Perivascular, by the way, means around the vessel, so that's why these are perivascular feet. Another view of the same thing. Here's a cross-section of my capillary. This is my epithelial cell, tight junction linking it. Then I got my astrocyte's feet hanging out around the vessel, providing a further barrier to anything bad in here getting out there. Notice we got some macrophages and protective microglial cells hanging out in the vicinity as well, just to, be on that protective lookout detail. Okay, one place we don't have a blood-brain barrier is at the choroid plexuses, because that's where we actually need things to leave the blood vessel, because we're making CSF. So we need water to get out of that capillary there. This is a choroid plexus capillary. Water's got to get out. That water will contain all sorts of stuff: glucose, oxygen, vitamins, important ions as well. And we'll pick up some wastes and unnecessary solutes too. So no blood-brain barrier when we are in the choroid plexuses. And wow, that third brain video was the shortest of the three, which I'm sure is cool with you. It is absolutely cool with me if I can, knock out a video in 18 minutes. I'd rather do a 30-minute one and an 18-minute one than a 50-minute video. Anyway, enough talking. I'll see you guys next time when we discuss the spinal cord. Bye-bye.